So my video is about the cell cycle, and it's about interphase, M-phase, mitosis, meiosis, cytokinesis, the checkpoints, and the crossing over. And so in the first part, we'll be talking about the interphase, which is where the cell spends most of its time. And it starts out with the G1 phase, which is where the cell's growing, and that's where it's doing its normal metabolic processes. And then in the S phase, after that moves to the S phase, and that's where each of the chromosomes are duplicated. They're made, a copy is made of each of them. And then in the G2 phase, it's continuing to grow and preparing for mitosis. So now we're going to be talking about the M phase of mitosis. And first, to start off with, we start off with prophase. And in prophase, the DNA is starting to condense, and it's made into distinct chromosomes. And then the prometaphase, that's when the spindle fibers attach to the chromosomes, and they're preparing to separate to go to the middle of the cell. And then in metaphase, that's when the spindle fibers are already attached to the kinetic cores and the centromeres, and that's when they go to the middle of the cell and start to divide in half. Okay, so now in anaphase, the chromatids move to the poles in anaphase, and then in telophase, the chromatids are at the poles, and the nuclear envelopes begin to reform, and then that's also when the chromatids decondense and the aster breaks down, and then after that cytokinesis begins. And cytokinesis is where the cleavage starts to form again. The cleavage furrow forms, and then the cell plates form when it's in a plant cell, and then they separate, forming two daughter cells, two diploids. And now we're going to go over the checkpoints that happen during mitosis. So the first checkpoint that we're going to start off is the G1S checkpoint. And this checkpoint, what basically happens is that it checks for DNA. It checks the DNA for damages, and it also checks to ensure that the proteins that are needed to carry on interphase and to go into mitosis are present. And the second checkpoint here is a G2M checkpoint right here. And this one is the checkpoint right before mitosis, and it's checking for MPF, which is the mitosis promoting factor. And what this is, the mitosis promoting factor, is it's a cyclin dependent kinase, and this is basically the proteins that it needs in order to go into to mitosis. This is what this checkpoint is checking for. Okay, so the final checkpoint is the spindle checkpoint, or the APC checkpoint. And what happens in this checkpoint is cohesion is being broken down. It's being broken down by separase, and separase has an inhibitor securin. So APC comes with another enzyme and destroys the securin, which frees up the separase to break down the cohesin, which allows for anaphase. Okay, so now we're going to go over meiosis. And meiosis starts off with one diploid cell and then ends with 4 non-identical haploid cells. So let's go over this. First, it starts off with prophase 1. Prophase 1, the chromosomes condense and the nuclear envelope breaks down and crossing over occurs. In metaphase 1, pairs of homologous chromosomes move to the equator of the cell, and anaphase, the chromosomes move to the opposite sides, the opposite poles. In telophase 1 and cytokinesis, chromosomes gather at the poles and the cytoplasm divides. And in prophase 2, the new spindle forms around the chromosomes of both cells, and metaphase 2 chromosomes lines up again in the equator. And then anaphase, the centromeres divide and the chromatids move to the opposite sides. And then in telophase 2 and cytokinesis, the nuclear envelope forms out of the two sets, and cytoplasm divides and the cleavage furrow, making 4 haploid cells. And crossing over occurs during prophase 1 meiosis, and it's when homologous chromosomes, tetrads, cross over. The exchange of genetic information, and this is what causes genetic variation in offsprings or in the haploid cells in meiosis.